Comes from the field of anesthesiology. That's where he still spends most of his time. I wished he spent more time with fascia research, but he tells me that the work he does is related to fascia, and now he can prove it. And he has become an assistant professor in the last year, and we are very proud. And he especially spent a lot of time looking at the effects of temperature on fascia. Thank you very much.
So in the next couple of minutes, I will talk about the physiological background, because my training basically is a physiology training. So I'm understanding myself as a muscle physiologist. And so I will briefly go into detail what a muscle physiologist can tell you about the myofascial system in terms of function and in terms of physiological background. So all we hear in this week is a lot of knowledge which has been produced by enormous amount of researchers all over the world, but I can assure you all we know about fascia is not more than the tip of the Ulmer Münster seen in the foggy winter weather of Ulm. There is a lot more to discover, and you are still at the beginning of a very interesting era. So I will talk a bit about biomechanics, energy efficiency, and then I will go into detail of facial contractures, a bit of fascial pain. And if you compare the cross-section of an orange with a cross-section of a tie, you will see a striking similarity. The fascial sheets basically give intersections, and they define the three-dimensional shape, and they define the three-dimensional appearance, the outer appearance of the whole thing. So if you would open up the orange and not knowing how to separate the compartments, you would have just water flowing around. Similar thing happens in our thigh muscle. Look that not only the muscle compartments are defined very well, but also the nerve and vessel compartments. If you look at this lady, I admit she looks a bit rusty, but you can still get an impression of her appearance, of her posture, and still you get an impression of her face. So this means that there is no water at all, so that's just connective tissue what you see, and you can still see that the fascia is a main component of the body shaping structures. In a more functional way, this is the classical assumption that the muscle does the work and the fascia is transmitting passively the force onto the joints, and the fascial components more or less have the same length, whereas the muscle tissue get longer and shorter. But recent studies have shown that this classical assumption is not valid anymore, because Kawakami did ultrasound investigations where he could definitely prove that the muscle almost contracts isometrically, whereas the fascia does all the elongation. So the field of fascia has changed several classical textbooks. Also the recent Olympic Games in London, where Oscar Pistorius was competing against athletes who have both lower legs. So, and he wears this lower legs prosthesis. He was a very competitive runner. So this again shows that the muscle tissue itself cannot be the only clue to a good performance. I want to add some data of Andrew Franklin Miller, and he showed that if you lift the leg and you put the lower leg tension to 100%, then you can calculate that 100% Achilles tendon loading and 103% of the lateral crural department down here. But what is surprising, that you load the lumbar fascia to 145% and you load the iliotibial tract to 240%. So you would expect that the lower side of the leg has the most load, but it's not the case. So this again supports Andre's findings and Achuton's findings that the lumbar fascia plays a major role in locomotion. So let me move on to the physiology of fascial hydration. So how can we hold water inside our tissue? The water content of a newborn is more than 80%, and in elderly people it can drop to below 50%. So there is a huge difference. How can the water be held into the tissue? So again, there are molecules which take part in this action, and the most prominent ones are the proteoglycans. Those are very, very large molecules which have a lot of branches, and they look more or less like a toilet brush, so they can hold water inside, and they can hold more or less water. And I'm convinced that manual therapy and physiotherapy changes the water binding capacity of those molecules. So we did an in vitro setup. So recently a paper came up which shows exactly our findings. We found that if you stretch a piece of fascia, you get a certain behavior, an initial peak, where you can get a peak force. In this case of this fascia strip, it's maybe 38 millinewton. And then if you let it rest for half an hour, you can find an even bigger tension, which is really 1.5-fold in this case. And so what is the mechanism behind? We were thinking of the active contraction, but we ruled out in these bundles because we did deep freezing. So those bundles reacted in the same way when they were deeply freezed and retort. So the cellular components went away completely. So we analyzed the water content, and we found out that if you stretch, the water content briefly after stretch, roughly 15 minutes after stretch, drops significantly, and after a while the water content refills, and the fascial bundle even can have more water content than it had before. This is even dependent on the strain amount. So in 4% strain it does not reach the level of significance, whereas at 6% strain the supercompensation is statistically significant. So what factors do affect fascial hydration? So we have seen in our previous data that the mechanical influences are therefore rehydration strategies, and there are other factors which are important: pH, temperature, and bioactive proteins. So the pH optimum is in a very narrow range regulated in our body, and if the pH drops then more than 10%, it is lethal. So it is a very important factor, and it is therefore so important because it's necessary for all kinds of biological reactions. All enzymes have a pH optimum where they need to work. So, and pH optimum also allows electromagnetic shielding of the cells. I grew up with Starship Enterprise and Starship Voyager, and most of you probably know that the starships, they have an electromagnetic shield to protect themselves. But nature is much more advanced than the Starship Enterprise, because you know that you remind yourself of a cell where inside you've got a lot of potassium, and outside you've got a lot of sodium. And if you want to activate the cell, in any case, you need the sodium to enter the cell. So there is electromagnetic sheath which is built by calcium and which is built by magnesium. And because those are bivalent cations, the sodium needs to pass the positive against positive. They need to pass this electromagnetic shield. And so if you have a good electromagnetic shield, your excitability of the cells will be reduced. Therefore, the pH optimum is very important. You know this yourself. If you've got a person who is hyperventilating, you will destroy this electromagnetic shield and you get cramps or muscle cramps. It's a neurogenic cramps, which has been shown recently. So nothing to do with the muscle cells themselves. So how is pH regulated? It's regulated in a very narrow range by basically two mechanisms. It's regulated by renal compensation, which acts within hours, and it's regulated by a respiratory regulation, which is working with every breath we take. And we can easily try to reduce our CO2 level in the body when we do a diving strategy, just to leave out some CO2 and to refill it afterwards. So this is the basic strategy, how we can control the dissociation of water, CO2, carbon dioxide, and protons and anhydride. So if I want to make the link again to fascial disorders, I want to give an example of panic disorders or breathing pattern disorders where the pH is chronically too high, which means that there is an alkalosis, which means the electromagnetic sheath is damaged, which means that there is a chronic hypocapnia. And so it happens that potassium leaks the cells in the so-called Hamburger shift reaction, which means, in other words, that we have a different biological surrounding for our fascia, and we can monitor this by observing clinically a higher incidence of joint hypermobility and mitral valve prolapse. So there must be something in the fascial surroundings which is very linked to the pH. And Pipil-Zadeh and Naylor, they found fascial contractures in vitro which are stronger in acidic conditions. And on the other way, they found that if you turn around the pH to more alkalotic conditions, you can even block those contractions. So the authors speculated that this might be a very important factor in wound healing. So if you have a wound, it's more or less acidic, so the pH is very low, the contraction is higher, so the wound can contract better. So that's a probable explanation. I want to spend in the temperature effects of fascia. If you look at a muscle bundle, this is a human muscle bundle of the gastrocnemius muscle. And if you check out the physiology of a normal muscle cell, you remind yourself that it is activated on the end plate by acetylcholine, then the T-tubule transfer into the muscle cell very fast, the interaction, and then you get an activation of the calcium efflux of the sarcoplasmic reticulum, and then you get an increase of calcium in the cell. By the way, this was identified in Ulm. Then you get a contraction of the cell. So if you look at it in an experimental setup, the right-hand side gives you the calcium indicator, the left-hand side just a microscopic view. And you see the calcium, there is a potassium peak, the calcium is increasing, the green color gets more bright. On the left-hand side, you see the contraction of the fiber. So this is how muscle works, as a fiber without fascia. So it is very much temperature dependent because it's a biological reaction, it's pH dependent. This is the same reaction as monitored as force versus time at 20 degrees Celsius. This is 30 degrees Celsius and this is 40 degrees Celsius. You can realize that there are two things. Muscle cell is reacting at a lower peak force and slower. And if you increase the temperature, it gives you a higher peak force and a faster twitch. So if you do a temperature versus time diagram, you can see that it is a logarithmic relationship. So exactly giving you the logarithmic law of biological reactions as you know it from biology course at school. So it works in daily life as well. So how does this go together with the fascia? Because you cannot divide epimysium, endomysium from the muscle cells themselves, so you need to see the whole bunch together. So how does fascia react in different conditions? If you look at the blue curve, which are the cold conditions, compared to the red curve, you see a higher peak force at the blue curve and lower peak force at the red curve, and you see that in cold conditions, the stiffness, which means the drop of force, is much more steeper decline here. If you look at clinical data, they show exactly the same, which has been shown by this group in the supraspinatus tendon. So again, they did a similar study, not in vitro, but in vivo, in the supraspinatus tendon, and they see that it's again the same pattern can be shown in vivo. So summarizing these effects means that at high temperatures, the stiffness of fascia is low, the peak force is low. At low temperatures, the stiffness is high, and the peak force is high. So if you look at the muscle, at high temperature, you get an increased muscle force, you get faster parameters of contraction and relaxation, you get an enhanced excitability of the muscle cells, you get basically activation of all processes. Whereas in fascial tissue, you get the opposite. You get a relaxation when you get heat, and you get a calcium-independent mechanism of the tension. So this means that usually you would expect that both systems act in the same direction, but exactly the opposite is the case. So how can we explain this from a physiological point of view? If you have a cold muscle, there is more tension given over to the fascia, and if you have a working muscle, then the fascia tension is reduced, and the muscle itself, the muscle cell, take over a more load-bearing situation. So let me go on to some other factors which might influence the fascial matrix. And these are bioactive proteins like the TGF-beta-1, transforming growth factor beta-1, connective tissue factor growth, or insulin-like growth factor type 1. This sounds very technically, and this can be shown in vitro conditions. However, what I want to explain with this slide is that if you know that cytokines are involved in inflammatory processes, are involved in our nutritional factors, because everybody who works in a lab knows that if you want to do cell cultures, you just put a bit of insulin. It's much cheaper than you put the insulin-like growth factor, because insulin is reacting on the same receptor, it's less specific, but it's working. So if you want to understand those factors, you need to step back and think, how do I increase all these factors? And these factors are enhanced by the thyroid glands, enhanced by the renal glands, and are enhanced by hormones like insulin and inflammatory processes. What about sex hormones? Because you have the obvious difference between male and females, so there must be some influence to our myofascial system. And indeed, estrogen receptors have been found in fibroblasts. The estradiol, as a hormone, inhibits the collagen synthesis, and estradiol inhibits fibroblast proliferation. So far, these are in vitro findings again. Let me move on to some clinical data. If you compare the incidence of low back pain of women who took, let's say, in former times, anti-baby pills, which had a high content of estradiol, they had a higher incidence of low back pain, of chronic pelvic pain, and pelvic joint instability. This is a proof that sex hormones play a role in the myofascial system. Let me move on to the fascial contractures. So this is a slide Robert produced in his research, which shows the lumbar fascia of a 19-year-old male. And he identified those stained areas as myofibroblasts, which are contractile cells within the lumbar fascia. So when you look at the cells, then you can find cells which are contractile and cells which are less contractile, meaning that there are fibroblasts turning into myofibroblasts by differentiation, and also potentially there is a way back that a contractile element can go back to a non-contractile cell again and rest until it's necessary to get contractile again. We did force measurements in our organ bath, which has very strictly controlled conditions, temperature, oxygen, pH, and so on, electrolytes, and so on. And Robert could show that mepyramine, which is again a drug in the histaminic level of hormonal influence, shows that there is a strong contraction in this sample. The pathophysiological background, there are two mechanisms by which myofibroblasts can be activated. There is a calcium-independent pathway, and this is obviously the dominant pathway because it's a Rho kinase-mediated inhibition of the myosin phosphatase. So this means that it is like a slip and ratchet model. So if once you activate, the fiber stays at the same position and you do not need any more energy. Whereas the calcium-dependent pathway is the more energy-consuming pathway. The cell does have both pathways and can use up both pathways depending on what is necessary for it. So the group of hints showed that fibroblasts have dynamic oscillations of fibroblasts of calcium, which are found usually in cardiac cells. So this is quite unusual for a cell to have calcium oscillations. And so this meets a specific background of all the messenger proteins to arrange a calcium oscillation in a cell. If you look at this cell, you can see that stepwise a magnetic bead, which is attached to the cell, has been translocated. So every time the calcium oscillation is there is a step up in contraction. So this means that there is not just a calcium oscillation, but the calcium oscillation has a mechanical answer. The pathology found in fascial tissue can be seen in several diseases, like the Poitrin's disease, Leatherhouse disease, frozen shoulder, and so on. In all of these pathologies, you find massive proliferation of myofibroblasts. And these conditions are very common, but how they are produced is not clearly understood. So I want to step back and show you how fascial remodeling is basically found in several other medical conditions. So for this, we need to look at the microenvironment. We already did. We looked at the interaction of the lumbar fascia, of the agonists and antagonists in the fascial string. But as Paul Hodges mentioned, we also need to look at the spinal cord, at the reflex circuits, and at the central nervous system. And if you remind yourself of basic functional anatomy, you remember that if this is the brain, there is one axon, so one cell going from the brain to the spinal cord, the next cell going from the spinal cord to the organs. So this is the very simplistic system which is still valid for many, many years. And you have exactly two cells controlling, a central cell and a peripheral cell. If the central cell does not work, the central axon is broken, you get a spasticity. And if the peripheral nerve is broken, you get a flaccid paresis. So this is a very simple thing, which is easily understood if you look at this example of the brain and the homunculus inside. So this area basically is the area of the middle cerebral artery. You can easily understand that arm and face are involved in this pathology. So if you treat patients with stroke, you will realize that it's not just a muscle problem they have. After a while, they have a remodeling of the fascial tissue. And so it is not a muscle problem you need to treat. It's a problem of the fascial tissue which you need to treat. Another thing which has not been mentioned already are all the neurotransmitters, which have a big influence on our movement. So We already got to know acetylcholine, but we got to know the peripheral acetylcholine, which acts on the muscarinic type of acetylcholine receptor, which acts on the nicotinic part of the acetylcholine receptor. In the brain, we've got the muscarinic type of acetylcholine receptor. So we have a lot of neurons which are cholinergic. We have biogenic amines which act in the brain. And if you look at serotonin, this is nutritionally impacted if you eat some chocolate. People say that serotonin levels can be raised and makes you happy, more or less. And drugs like amphetamines acts on serotonin levels. Then we have dopamine, which is very much linked to our movement, which is linked to thinking abilities, and which is linked to our mood. So look at little children. They are in the kindergarten, run around all day long. They have a high amount of dopamine inside. They have a good mood, they are happy, they run around, they have loose connective tissue. So those children have a lot of neurotransmitter in their brain. So this is an important factor as well. On the other side, if you look at psychiatric patients, if they get antipsychotic, anti-neuroleptic drugs, which regulate down their thinking processes, they walk like a robot because dopamine receptors are blocked. Neuroleptics do nothing else, just blocking dopamine receptors. So what you have, a bunch of neurotransmitters which are very important in our movement and in the development of fascial remodeling, because you need neurotransmitters for keeping mobile. So let's move on to the amino acids. Glutamate is the one you know from the Chinese restaurant when you get a headache after having too much Chinese food. You also probably know gamma-aminobutyric acid, which is the main component in all tranquilizer drugs. Then you have neuropeptides like endorphin, which relieves pain, or on the other side, which substance P, which increases pain. So those are important for fascia as well. Recently, gaseous neurotransmitters have been found, even in the brain, and this is nitrogen monoxide, and in our lab we already tried to use those transmitters and to use up the NO donators for our experiments, but the results are not consistent yet, so probably the next time we can present data on this. When you look at the classical model of back pain, for instance, you have the neuromuscular control unit, which is brain and spinal cord, which I showed you already, and then there is the spinal column as an organ with all these factors like the facet joints and the vertebrae and so on. And so the classical assumption says that this is an interplay and that the spinal column is the primary origin for low back pain. However, this classical assumption is not true anymore, since we know that the fascia plays an important role in pain generation. So look at this cross-section. You can easily see that this is the spinal column. These are the fascia surrounding the posterior muscles and the medium muscles and the psoas muscles. And if you look at the lever arm is much smaller to the facet joint than it is to the fascia. So it means that much more tension is transmitted to the fascial components. So I want to conclude by applying the physiological knowledge we already have to long distance runners. And a friend of mine, he went with a large truck. He followed long distance runners with this MRI truck, and this was ultra marathon in 2009. And so what I want to show you is this MRI scan of day one. So this is day one and the MRI scan is on the calf level. So what you see basically the bone structure, you see the connective tissue, which has a white appearance, and you have the muscle tissue, which has this dark gray appearance here. So if you look at stage 25, so it's after 1,700 kilometers, so you readily see that at different levels of the calf, you have an increase of signal in the fascial segments, you have an increase of signal at the outer area of the fascial segments. So not only inside the calf, there is an increase of connective tissue, but also outside. So this thickening can be seen as an inflammatory disease, and on the other hand, you get muscle wasting, so muscle bulbs getting smaller. If you look at the clinical picture of those runners, you will see this reddish appearance. And the reddish appearance is not because the skin is inflamed, but because the fascia is a main component of load bearing and elasticity. The fascia is inflamed itself. To conclude, there were 44 subjects starting, roughly 70% were finishing, and it's interesting to note that among the non-finishers, the most important reason for signing in, for giving up, is leg pain, is soft tissue problems. So again, obviously our connective tissue plays an important role in long distance running, in very high endurance situations. So I want to conclude with this headline, with this title from a recent article on fascia in Men's Health, stating that everything you know about muscle is wrong. Thanks for your attention.